Hello my dear students, today we are going to discuss about the reproduction in bacteria. You know that there are two types of reproduction, one is asexual and second one is sexual reproduction. Today we will briefly focus on the asexual reproduction in bacteria. First of all, what is reproduction? Brief overview. Reproduction is a biological conduit for the transfer and perpetuation of gene pool down the generations. That means it is a way of transferring the gene pool, transferring the genetic material to the new generations. Okay, or simply stated, it is a means of perpetuation in living organisms to continue their generations, to continue their existence. It is of two broad modes. One is asexual reproduction and second one is sexual reproduction. In asexual reproduction, what happens is single parent is involved, there is no genetic recombination. While as in case of sexual reproduction, what happens is two parents, they participate in the sexual act. Okay, and this involvement of two parents, it favors, or you can say facilitates, the recombination of genetic material. Okay, that is known, that is genetic recombination happens in sexual reproduction. Now we will have a brief overview of the asexual reproduction and its different types. In asexual reproduction, a single bacterial cell participates in the generation of offspring. That means एक ही bacterial cell participate करती है reproduction में, and this cell forms the daughter cells without the formation of sex cells, meiosis and fertilization. This is very much important. You have to remember this. In asexual reproduction, a single bacterial cell participates in the formation of new offsprings, and this happens without the formation of gametes, without meiosis and without fertilization. Okay, no recombination of genes takes place in asexual reproduction, and the daughter cells which are produced, they are genetically identical. When recombination hasn't taken place, okay, recombination नहीं हुई है, तो जो daughter cells बनेंगे, they will be identical to each other as well as identical to the parent cells from which they were formed. And these identical daughter cells, they are known as clones. They are known as clones. Asexual reproduction is done in case of bacteria by various methods, or you can say asexual reproduction employs a number of methods. The very first one is binary fission, then we have budding, then Conidia formation, then perinating buds. It includes cysts and endospores. Okay, so first of all, we will discuss binary fission. Binary fission is the simplest and most common type of asexual reproduction, and it occurs under favorable environmental conditions. When bacterial cells grow in favorable environmental conditions, when nutrients are in abundance, when temperature is optimum, when humidity is optimum, at that time instant, the bacterial cells, they prefer to go for binary fission. Okay, binary fission होता है क्या actually? Binary fission में one cell, it forms a septum and forms two cells. Okay, in binary fission, what happens is, first of all, cell elongation takes place, and then diffusion of genetic material occurs, which is followed by diffusion of cell protoplasm into two daughter cells, and each daughter cell receives one copy of the nuclear material. यहाँ पे आप देख लीजिए, say for example, this is a bacterial cell, it has this nucleide. Okay, what happens is, this cell first of all elongates, it increases in size, its genetic material divides, it replicates and forms two copies. Okay, then this cell, it divides by a septum. Okay, and each cell gets the protoplasm, it gets the genetic material. Okay, now some important remarks about binary fission. Binary fission is extremely rapid. Okay, this process takes place almost every 20 to 30 minutes. After every 20 to 30 minutes, each bacterial cell divides to form two cells. However, it may be constrained by limited nutrients and accumulation of wastes. When nutrients become scarce, there is scarcity of nutrients in the environment and there is accumulation of toxic materials in the medium. The bacterial cells, they stop binary fission, okay, or you can say the pace of binary fission, it slows down. Unlike normal mitotic division, in binary fission what happens is no spindle formation occurs during division of genetic material. याद रखना, this is very much important. In binary fission, there is no spindle formation, okay, during the replication of genetic material, while as in case of normal mitotic division, spindle formation happens. The daughter cells are an extra replica of the mother cell. The daughter cells, they are an extra replica of the mother cell. That means this is mother cell, you have two daughter cells, they are the extra replica, okay. That means with every single instant, with every single division, the number of cells increases and they are exactly similar to the parental cells. It is characteristic of all bacteria. Fission occurs in all the bacterial classes and groups, and hence the bacteria, they have been classified and placed in a single class named as schizomycetes, or you can say split fungi, okay. Fission is characteristic of all bacteria, hence they have been named as schizomycetes, okay. Here you can see this is the diagrammatic representation of the binary fission. This is the parental cell, it increases its volume, okay, and it is genetic material, it divides, forms two copies, or you can say it, DNA replication happens, it forms two copies. Then septum formation takes place here, it begins here, it initiates here, then finally the septum, it grows inwards and the two dot cells are formed, okay. This is the simplest representation of the process of binary fission. Another mode of asexual reproduction in bacteria is budding. First of all, what is a bud? A bud is an outgrowth arising out of the parent bacterial cell, okay. For example, this is a bacterial cell, an outgrowth arises from this bacterial cell. This outgrowth is known as bud. In some cases, multiple buds from a single cell have also been documented, okay. कुछ instances में देखा गया a single bacterial cell can have many buds, okay. A single bacterial cell sometimes produces multiple buds, and these buds, they are filled with cytoplasm and other cell organelles. जो ये buds होते हैं, they are filled with cytoplasm and other cell organelles which are present in bacterial cells. When the buds become mature, they eventually detach from the parental cell and they give rise to new daughter cells, okay. Say for example, ये वाला bud यहाँ पे detach हो गया from the parental cell here. This bud will grow in size and it will form a new bacterial cell. Now the examples of bacteria which undergo budding include Hypomonas, Labries, Stela, and there are many other bacteria which undergo budding. Here you can see this is the diagrammatic representation of the process of budding in bacteria. This is the parental cell, and this parental cell initiates the formation of a bud here, okay. Then what happens is this bud increases in size and the nuclear material and the cytoplasmic contents, they start migrating to the bud. After some time interval, the bud starts separating from the parental cell, and after separation, okay, it can give rise to new bacterial cells that we have discussed. But here in case of this diagram, you can see this bacterial cell has not produced a single bud. Instead, it has produced a chain of buds. Okay, and these buds, which are present in the form of chains, they can later on detach and give rise to a number of bacterial cells. One more mode of asexual reproduction in bacteria is Conidia. Conidia formation is characteristic of filamentous bacteria. ये याद रखना. Conidia specifically forms in filamentous bacteria. Okay, these are reported in those bacteria which are filamentous, or you can say whose bodies are filamentous like fungi. What is conidium? First of all, conidium is a small spherical spore-like body formed at the tip of the filament, and this conidium is demarcated, or it is differentiated, or you can say it is it becomes different from the filament by the development of a transverse wall at the tip of the filament. Okay, say for example, this is the filament, this is the tip of the filament. Okay, a transverse wall happens here, a transverse wall forms here, and this transverse wall results in the formation of a spore-like body at the tip of the filament, which is demarcated by a transverse wall. This structure here, this is known as conidium. A number of conidia forms successively at the tip to form chains of conidia. In some cases, you will find not only a single conidium is formed, but a chain of conidia are formed successively, and they result in the formation of chains of conidia. Okay, and the filament which bears these conidia in chains, this is known as conidiophore. This filament is known as conidiophore. Okay, the filament bearing conidia, this is known as conidiophore. Just like the buds, the conidia, they eventually detach from the filament and germinate into new filamentous bacterium under favorable environmental conditions. Here you can see this is the diagrammatic representation of conidia formation in bacteria. Here you can see this is the conidiophore, or you can say sporangiophore. Okay, this conidiophore, it bears chains of conidia in succession. Okay, they are attached to each other, and these conidia after maturation they detach from the conidiophore and they disseminate to various places, and each of these conidia, they later on germinate and give rise to new bacterial cells. Okay, I think this is all for today. See you soon with the remaining modes of asexual reproduction in bacteria. Till then, goodbye, take care.